All right, hello everyone. This is chapter 4, cell structure and function. We're going to be talking a lot about how structure determines function here. I love this meme. If you don't know what structure determines function is, I would definitely review that because we're going to be talking a lot about structure and function in this chapter. All right, first, let's talk about the cell theory. Cell theory, all organisms are composed of cells. First part of the cell theory. Cells are the structural and functional unit of all living things. That just means the most basic and smallest thing that we consider a cell or that we consider living is a cell. Third, all cells must come from pre-existing cells. 3 parts of the cell theory. You should have heard those back in regular biology, but just know those main 3 parts of the cell theory. All right, prokaryotes versus eukaryotes. This first chapter is going to be focused on eukaryotic life and eukaryotic cells, but I still like to go over the basics between the two. Prokaryotes are bacteria and archaea. There's no nucleus. They lack membrane-bound organelles. They're less complex. They're smaller. There are billions of prokaryotes in your mouth, around your body right now, and just everywhere. They're very, very small. Eukaryotic cells. These are much more complex. These include plants, animals, protists, and fungi. They have a nucleus. They have membrane-bound organelles. They have inner working organelles. They're much more complex and they're larger. So that's the main difference between both of those. Here's a good image of a eukaryotic cell and a prokaryotic cell. Again, you can fit hundreds, if not thousands, of prokaryotic cells in a eukaryotic cell. The first organelle that we're going to go over is a ribosome, and your cells have like thousands of ribosomes in them. Again, we're going to go over both the structure and function of a ribosome. You can see structure made of 2 smaller subunits. Now, the ribosome is made of protein and rRNA. We'll learn more about this later on throughout the year, but the main thing to remember is the ribosome has 2 small subunits that go together to form this ribosome. In like regular biology, you learn that this is like a dot in the cell, but there is actual structure to these ribosomes. Function produces proteins based on DNA and RNA sequences. That's going to be transcription and then further on translation. But these proteins are going to be what makes up your body. Now, the cells. All cells have ribosomes. Every single one of them. Every single one has ribosome. It doesn't have a ribosome, not technically a cell. You need ribosomes because you need to build protein. So you need these ribosomes. Okay, now next, a cell will also have a phospholipid bilayer or a plasma membrane. And these phospholipids we talked about before. We talked about how these phospholipids separate the outer matrix from the inner matrix. These phospholipids also just regulate what goes into and out of the cell. So every cell will have a plasma membrane. All cells will have ribosomes. We have one more to go over. Oh yeah, membrane proteins aid in regulation. We actually spend a lot of time talking about these membrane proteins next chapter, but yeah, these membrane proteins are going to be what regulates and helps regulates the passage of things into and out of the cell. Last one here, the cytosol. This is the semi-fluid-like solution that is composed of, or what's in the cell. It's sometimes called the cytoplasm. The cytoplasm is everything together. The cytosol specifically is the liquid portion inside the cytoplasm. There is a difference between them, although they are used interchangeably. But function, it helps hold other organelles in place. And there's a lot of other molecules in the cells, not just this liquid. There's a ton of molecules we're going to be talking about. So the cytosol is that semi-fluid solution inside the cell. Here's a great comparison between the two. Cytosol, this is that fluid within the cell. The cytoplasm is absolutely everything within the cell. Okay, so to end this is a good meme. Eukaryotic cells and prokaryotic cells, the 3 things they have in common: plasma membrane, ribosomes, cytoplasm. All right, 4.2, the endosymbiotic theory, one of my favorite topics to teach about all year long. So basically, we're going to talk about how eukaryotic cells became eukaryotic, because now we have some fundamentals of all cells. We have a plasma membrane, we have cytoplasm, and we have ribosomes. But how do we have these different types of cells, the eukaryotic versus the prokaryotic cells? So one thing that we see here is that mitochondria and chloroplasts are in these eukaryotic cells, and these are membrane-bound organelles. And they're very specific membrane-bound organelles. What the evidence points to is that over billions of years ago, there was a huge evolutionary advantage for a cell, as an ancestral prokaryotic cell, to intake other cells. Now, you can see here it says cyanobacteria and it says aerobic bacterium. These were their own living cells. These were not just like, these were living cells. This, for whatever reason, this prokaryotic cell folded in the outside plasma membrane and took right out of the English, the cyanobacterium and the aerobic bacterium and kept them around. Instead of killing them, they said, hey, these cells, if we keep them around, make glucose really well, make carbohydrates really well, and they break down those carbohydrates and they produce ATP very well. So these organelles that are membrane bound were once free living organisms that we took in, eukaryotes, billions of years ago. And you're like, what's the evidence for that? Let's go over the evidence. First, they have their own membrane. There you go. First, they have their own membrane. They have a double membrane, actually. The outer membrane more resembles that of the outer one resembles that more of their eukaryotic membrane. The inner one resembles more of a prokaryotic membrane. That's because, again, you can see the outer membrane enclosing this inner membrane. Yeah. So they have two membranes. Next, DNA. So all of your DNA is in the nucleus, right? Wrong. Your mitochondria and chloroplasts in plants have their own DNA. They have their own DNA outside the nucleus. Another interesting thing about that DNA is it's circular. And if you remember, what type of cell has circular DNA? And that's prokaryotes. Interesting. Ribosomes. They have their own ribosomes. And those ribosomes they are more resemble those of prokaryotes. There are differences between the like how they make up the ribosomes, but yeah, the ribosomes that you have in your cells are also found in the mitochondria and chloroplast. And again, they resemble more of the prokaryotic ribosomes. Reproduction. They also reproduce very similarly to prokaryotic cells. They have very close to asexual fashion of splitting. And lastly, and this is the most subsequent, not really important, but still there, size. They're the same size as prokaryotes. So yes, these bacteria or these organelles were once bacteria that lived billions of years ago that eukaryotes just took and said, hey, I'm just going to keep them around. We can see that the mitochondria is in all eukaryotes, the animals, fungi, and plants here. And then we can see that plants are the only ones that took in, the English is not the greatest today, that took in the chloroplasts. So all plants then have those chloroplasts. That's why we don't have chloroplasts. All right, the nucleus. Let's go back one. Oval, found near the center. It's a structure. Its function is contains and protects all the DNA. It also contains chromatin. These are strands of DNA. And let's make sure we understand what that looks like. Oops, go back. So this is what the chromatin actually looks like. It looks like a bunch of spaghetti. But make sure you understand that the chromosomes aren't condensed yet. Most of the time in a cell, they are not condensed. Only in a small portion of cells do we see the DNA condensed. It's usually very I should say free floating. It's unraveled. Yeah, it's just a jumbled mess. Okay, and that makes it easier to access when we need it for transcription and translation. But we're not there yet. But that's what a nucleus is. Oh, before I leave here, nucleolus. This is the dark region within the nucleus. This creates something called ribosomal RNA. And that's, if you remember, what makes up ribosomes. All right, the last slide in section 4.2. The nuclear envelope. So this is the outside of the nucleus. This is the double membrane that we find. This is what protects the DNA and is a kind of like the regulator of what goes into and out of the nucleus. Nuclear pores. These are the sections or the little holes within the nucleus that allows for the passage of rRNA and the mRNA to leave and go back and forth between the cytoplasm. Now, both of these structures together regulate what goes into and out of the nucleus. All right, chapter 4.3 is another favorite section of mine. This is going to talk about the endomembrane system. Thinking endo, meaning in, membrane, thinking that phospholipid, and system. We're working together between these different organelles. The endoplasmic reticulum, also known as the ER, is a complicated structure of membrane channels that's going to be surrounded by the nucleus. Well, it's surrounding the nucleus, not surrounded by the nucleus. Yeah, so what happens is there are two types of ER. You might remember these from your 9th and 10th grade biology days. You have rough ER and you have smooth ER. Let's talk about the difference between the two. Rough ER, this is a structure that has ribosomes attached. So when you think that, remember back what ribosomes do, they make proteins. You can see your function here produces proteins, folds extracellular proteins inside the lumen here. You can see the rough ER around the nucleus. Next, we have the smooth ER. This is still connected kind of to the nucleus, but more through the rough ER, but it doesn't have ribosomes. So this is going to be more for making lipids and other molecules, and they are going to be involved in transporting vesicles to the Golgi body. Let's talk about that here in a second. Remember that the smooth ER doesn't have ribosomes, the rough ER does. So rough ER makes proteins, smooth ER makes lipids, but together they do make molecules that the cell is going to need. Here's a great little animation. You can see those vesicles leaving for the Golgi body. We'll go over both of those terms here in a second, but you can see those vesicles with proteins and lipids. You can see again, the vesicles leaving the Golgi, or leaving the rough and smooth ER. They're going to the Golgi body, and they're going to fuse with that Golgi body. I love that animation. All right, the endomembrane system, vesicle. These are just like little transporters the cell is going to use to transport things around the cell. They typically leave the rough and smooth ER to go to the Golgi body, or they actually will sometimes fuse with the membrane, and they'll expel materials outside the cell. They are made of a phospholipid bilayer, and yeah, they're just like a transporter of the cell. You can see a vesicle form. This is an actual visual of a vesicle form. You can see that plasma membrane forming that vesicle. Here are some images. So these are not real images, but these are artist renditions of a vesicle. So you can see the vesicles here, the vesicles here. Next, Golgi body. This is a stack of curved, they're called saccules. These are flattened vesicles. Basically, what this organelle does is it receives these vesicles from the ER. It modifies, ships proteins and lipids all around the cell. So this is like our UPS center. It intakes the proteins and lipids, and then it pushes out these vesicles to go to different portions or different places throughout the cell. And sometimes they actually leave the cell as well. So this is just mainly our, not really our control center in the sense of it doesn't have DNA, but this is our center in the cell that's going to really transport things around the cell. It's going to direct those vesicles of where to go. There's a lot of videos in here that I'm not going to show. I'll show in class. There we go. 4.3 is done. Section 4.4 is going to be looking at other membrane-bound organelles. Remember, when it's a membrane-bound organelle, that just means it has its own membrane, at least one membrane, if not two. Lysosomes are next. These are going to be membrane-bound vesicles that are produced by the Golgi body that also contain digestive enzymes. These are specific to breaking down like worn-out molecules. They're specific to breaking down like bacterium that are trying to infect the cell. Anything that the cell doesn't want around, these lysosomes are going to take care of. So what they do is they take in whatever the cell doesn't want, they break it down, and the cell either reuses that material or it just pushes it out and gets rid of it. So you can see there, yeah, the function destroys non-functioning organelles, invading pathogens and other content in the cell that is just not needed anymore. There's going to be a couple slides here that I'm not going to go through because there are a bunch of videos and a bunch of examples. Yep. Okay, peroxisomes. These are very similar to lysosomes. They have their own membrane. That contains enzymes. These peroxisomes help break down alcohols, formaldehydes, and other dangerous chemicals. The difference is these ones specifically detoxify the cell. So we're going to have different ions and different like molecules that will sometimes build up as a result of different reactions. And if your body allows itself to build these molecules up, bad things can happen. For instance, hydrogen peroxide can build up in your cells. So what these peroxisomes do is they break down those free radical molecules or atoms or ions or whatever, and they make them into useful things like water, oxygen, things that we can use. So these are really an important type of organelle because it really detoxifies the cell. Vacuoles are our next membrane-bound organelle. Again, remember, they have their own membrane. These are just like the storage containers of the cell. They're larger than vesicles, but they contain food, they contain liquids, they contain things the cell is going to need over time. And again, the function, they store things for the cell. All right, membrane-bound organelles. Chloroplast, another membrane-bound organelle. This is, again, remember, it has two membranes. This is from that endomembrane system, the endosymbiotic theory. But basically, this is an organelle filled with pigments that's going to intake light energy and make carbohydrates. This is the location for photosynthesis. We'll learn a lot more about these later on. Mitochondria. These are, again, another organelle is a result of the endosymbiotic theory. Double membrane, smaller than chloroplast just by a little bit. Function, they break down carbon molecules like carbohydrates and lipids to create ATP. This is the site of cellular respiration. By the way, please don't call these the powerhouse of the cell. Not the greatest representation of what these do. Yes, they do create not power, but energy, but they produce ATP by cellular respiration. That's a pet peeve of mine. Plant and animals, difference between these two. Plants, they have one large central vacuole. They have chloroplast and mitochondria. They have a cell wall. They're rectangular in shape, and they don't have lysosomes. How are they different than animal cells? Animal cells have many small vacuoles. They have mitochondria. They do have lysosomes, and they're more circular or irregular in shape. All right, the last section here is going to be on the cytoskeleton. I know I don't have it up here, but it'll be on the next slide. Basically, the cell wall is a structure outside of the cell membrane that holds and supports the cell, but also protects the cell. We only see this in plants compared with animal and plant cells because obviously animals don't have these. We can more freely move, but plants gain a really big support structure that they can use, and that's why they grow, hundreds of feet tall sometimes. But yeah, this structure, the cell wall is made of carbohydrates and proteins usually. But yeah, cell wall. Cytoskeleton in general is this network of protein fibers that's going to be found throughout the cell. And its main function is to support the cell and to move things within the cell. The green structure within the cell here, you can see these little fibers. This is the cytoskeleton. And there are a lot of different types of cytoskeletal structures, but I just want to show you the complexity here. There's a lot more going on in the cell than when you look at it just like an image of a cell. There's a lot more going on here. Big keynote, dynamic structure. This rearranges itself as needed. So once it's built, it doesn't just stop and just be there for a lifetime. There are a lot of ways that your cells reworks the cytoskeleton for when it needs things. All right, 3 different types of cytoskeletal structures I'd like you to know. Microfilaments. These are the smallest. They contain fibers of monomers called actin, and they have sometimes myosin motor proteins that run along them. The function is for support and movement. We're going to see that support and movement here in a second. You can see the myosin and actin working together. This is how your muscles move. These are motor proteins that will run along the actin molecules, and this is, again, how you move. This is how your muscles function. The intermediate filaments are in between the size here. They're a little bit larger than the actin filaments, but again, they're a rope-like structure. They're a protein. They function closer to the nuclear envelope and plasma membrane, but they're more static than the actin filaments. Example of one of which it would be keratin. Again, these are for structural for purposes to help support the cell. Lastly, these microtubules, these are the largest of the 3. They're hollow, composed of proteins called tubulin, and again, they help move things around the cell. So all 3 things help with the support, and all 3 things help with moving things around the cell.